All right, I'll kick us off. Thank you everyone for joining today. We're really excited to see you all here and talk about biological responses to ossification, studying physiological responses to meta-analyses. I think this is a great complement to Dr. DuPont's lectures this week about running biological experiments. We're very lucky to have a special expert here today, Alleluia Taisi, to talk about some of her work studying organisms that are really important to the Pacific. So with that, we'll kick us off. Alleluia, do you want to get started?
Yes, I will. Hi, everyone. Thanks for having me.
Do you
I'm
want to
not
provide?
expert.
You are. Do you want to provide just a little bit of introduction on your background?
Yeah. So hi, everyone. I'm Luya. My name is Alleluia Luya. I'm a student here at Victoria University of Wellington here in New Zealand. So I'm originally from Samoa, and I came from Samoa 3 years ago to study Kalerpa in terms of their physiological responses to climate change, right? And so my plan at the start was to learn how to run an experiment, proper experiments in that case, and learn about, how to measure these physiological responses and do all that. And then later on during my thesis, I'll take that to Samoa and run some similar tests in Samoa using Kalerpa in Samoa, right? But because of the pandemic, I couldn't do that anymore. So I ended up doing all my experiments here in New Zealand using Kalerpa that are commonly found here in Wellington. And so I have completed 4 lab experiments during this time, so mostly manipulative lab experiments. And my interest is on Kalerpa, the sea grape, so different types of sea grapes. So that's me learning,
That's
still learning.
fantastic. We're all still learning. No, it's great that you've had so much experience doing biological experiments, which is one of the things that we're really excited to hear about you or hear about from you today.
Yeah, okay. I will share my screen. Thanks, David.
Perfect.
Is everyone seeing that?
We see the notes on the side too.
Right. So if I do that, how's that?
Looks great.
Yes.
Okay. All right. So this is a combined talk with me and Caitlin, and we're just talking about biological responses to ocean acidification, from physiological responses, how to measure, how to run this experiments, at least from our experience, and then to meta-analysis, where Caitlin will be talking more about. So I guess I wanted to start off with, talking about manipulative lab experiments, like how to run lab experiments. So I think for me, what I've learned is that it's good to have some expectations. It's good to know what you are expected to do if you are interested to study an organism in the lab, right? So what are the expectations? What do we need to do? What do we need to measure? For example, how do we measure these things? So I guess in the next few slides, I'll just share with you what I have learned along the way. Obviously, we don't have all the time to talk about everything that we could talk about, but I'll just briefly touch base on some of the things that I think is important when running the experiment. And I guess it's also important to have some resources to read off from, so do some literature review and do some reading before carrying out a lab experiment, to see what methods are available out there, for example. So mostly in this next few slides is about manipulative or manipulation lab experiments. So to start off with, I think it's a good reminder for us to see and to know what the components to an experiment are. So I think we are all aware, and we know that, first thing that's very important is our research question and our hypothesis. And that's really, really important to be as precise as we can possibly be in our research question, because that will inform our experimental design. And I think in terms of experimental design as well, before that, we also have to identify what our key driver, what the driver of interest is to us. So I think you guys are also familiar with that. So, a key driver of climate change that is relevant to our situation, to our local ecosystem, to our local community, to the local species that are there. So for example, if we're looking at corals, so obviously the driver will be CO2, right? So higher CO2, pH goes down, and that'll not be good for the calcifying organisms. So we have to identify what are the drivers that are of interest and what is the key driver out of all the drivers that you can identify. Then you formulate your experimental design. And in the design, I also found that it's just not designing your experiment. There's a whole lot of logistics and planning and strategizing, in that phase. So this is where your good planning comes in, and identify what you have, what you don't have in terms of resources, in terms of space. Do you have enough space in your institution to run an experiment? How much can you afford, the costs and things like this. So that all comes, I think, under the experimental design to the execution part of the experiment. So I've highlighted the execution part as something that I wanted to talk a little bit more about in this talk. What's also important as a component to an experiment is the statistical analysis, and I think that's a whole lot lecture for next time, I guess, by a statistician who's an expert on that, but that is also important. At the end of our experiments in the lab, when we have all our data, how do we statistically analyze them? Once we statistically analyze them, then we interpret, of course, right? And then we report and tell a story from what we saw in our experiment. So as I said, I wanted to just go over the execution part. So in my mind, execution is, just bring this down here, is more on the, so I think there's like 3 phases in my mind from what I've learned. So the preparation and setup, as I said before, again, we're looking at our research question, understand what we want to find out, what we want to learn, and then we identify our drivers of interest, whether it's a global driver or a local driver. So drivers can be drivers of ocean acidification, and that's CO2, a driver of warming, and that's temperature, and things like this. The points that are highlighted here in the first box under preparation and setup, I think they are a little bit important, more important when we are planning for the experiment. It's the manipulation method, it's the treatment levels, and the replication. So I'll go through that in the next slide. The next phase of the execution part, I think, is what to measure, right? So now we know, okay, we have experimental design, we know our research question, we know our driver, okay, you want to do this, you want this. So what do we actually want to measure, right? So that means, what is the biological response or the traits of that organism that is of interest to you, or that is of relevance to your environment, to your community, to your coastal ecosystem, or things like this. An example is, we want to find out the survival rate of an oyster, of a baby oyster or something. It's like a juvenile or something. We want to find out how ocean acidification affects the growth of seaweed. So we want to measure the growth response, right? So these are some of the examples, and there's so many things that you can measure if you know you have your experimental design in place and your setup in place. So once you know what biological response, what do we actually want to measure from the organism under the experiment, then we also had to do some research and reading and find out a little bit about what methods that we can employ in order to be able to measure that response? How do we measure it? The next important phase in the execution part is when the experiment is running in the lab. What are we monitoring? What do we need to monitor? And this is the thing that really, like, I wasn't really expecting when I started to do some lab experiments and learning, how to do these things in the lab here, is the amount of physical labor time that we have to go into during these experiments. Not that this, to discourage everyone, oh my gosh, we're going to be in the lab forever, but it requires us to have a better planning, like have a daily checklist plan of things that you want to do. So what we need to monitor, for example, once the experiment is running, let's say we're running an ocean acidification experiment, the things that we have to keep an eye on are the water chemistry parameters, of course. So we have to make sure that we're measuring the pH every day or every other day. So that makes sure that our treatment levels are right at the range where we want them to be. And we also important for us to monitor the total alkalinity of the water in the experiment. So we might need to do some titrations every day or every other day. Again, it depends on the number of people in your group or your time and how you sort out your time. The other thing that we need to monitor is the flow rates. Okay, so the water that's running into your tanks, and I'll show you a picture in the next slide. We have to measure the flow rates because there's some studies that have suggested that the flow rates and the mixing of the water actually also affects some organisms in some parts of their ecosystem, right? So there's different water mixing, water movements, flow rates at some, it's different, like 3 meters compared to 5 meters, for example, for the organism living in 3 meter and 5 meter of the same species, they could be responding to different flow rates. So we had to also find out from some literature if someone had done that before, see what sort of flow rates is suitable for your organism. Also important to monitor the temperature in the water and the light levels in the water. Light levels you can measure every week, or every other week. So you can make sure that the light levels in the experiment is about, the same, and it's not like changing and we didn't know that, and therefore it's introducing another error into our experiment. Again, we had to do daily checks. We had to go around the experiment setup, check all the pumps, check that the heaters are working, check that the air pumps are working, check that the tubings that carry the water are not blocked and that there's water going into the tank. So these little things that we have to monitor. And it's good to have file notes and things like that to keep you on track of what's been happening each day during the experiment. I think I also don't have it down here, but now I remember in terms of preparation leading up to monitoring is the time, like how long do you think you want your experiment to last in the lab, right? So you might be looking at the responses of corals, for example. So you want to bring corals in the lab, manipulate the conditions of the water and the environment in the lab. How long do you want to have that experiment for? 2 days, 3 days, 5 days? So again, you have to, understand a little bit about your organism, biology, things like that, physiology, before you start the experiment. In terms of manipulation, this is so important, and I think it's one of the things that I really picked up from my experience with conducting this lab experiments is the manipulation method. So for example, for our ocean acidification experiment, what do we need to control, like what factors or drivers that we want to control, and what is our key driver that we want to manipulate. So obviously for ocean acidification, we want to manipulate the pH, right? So we had to identify, okay, how do we manipulate the pH in the water? What is the what is more suitable to change the pH in your water? Is it from a CO2 gas bottle, or is it from hydrochloric acid that is injected into the water to go into the water where your organisms are? So these things like this, it depends on your organism. It really depends on your question, and it really depends on the organism that you're working with. So for example, for me, because I am working with Calurba, fleshy seaweed, and also the response that I want to measure is their dissolved inorganic carbon physiology. So I think CO2 gas is more suitable for my organism, and it may not be suitable for maybe diatoms or micro algae or something. So you have to just find out what is more suitable to your organism. The other thing that I think for ocean acidification that had to also look at manipulating is the pH variability. So this is something that I picked up as well, where some of the studies in the past on ocean acidification impacts, they didn't really tell in their article, in their findings, in their research, or it wasn't really clearly mentioned, is how the pH was manipulated in terms of the variability. So I just wanted to bring to your attention again, and I'm sure all of you have know this already, that when we are running a lab experiment, we are bringing the natural environment of the organism into the lab, and so we have to make sure that we mimic the conditions in the lab to almost be the same to the condition in the field where the organisms are coming from, so that gives us a true indication at the end of their response, right? So for example, here in our site, so it's close to a kelp forest, and so there's really high variability in pH in this water that's coming into the lab. So the thing is, what we did with our experiments on ocean acidification, and for all of our experiments, we manipulated the pH of the water to change in a daily cycle manner, right? So we have at the start of the day, we have a low pH, and then it goes up as the day goes. So by midday, the pH is really high because all the organisms are photosynthesizing, and then it comes down again, and then it repeats. So some studies didn't do that from literature, and they just kept the pH constant. And so that also kind of could be affecting the organisms in your tanks, because some organisms are not now, they're in the tanks and they're not experiencing the natural variability as they usually experience out in the field. So they will respond differently because of the constant pH and not because of the actual treatment, right? So I think the manipulation of your conditions in the lab is really important. Also with photosynthesizing organisms, you might also want to consider, having diurnal cycle of the light levels in the experiment, because the light in the field is also changing, right? So in the daytime, there's high wavelengths of light, and then at night, there's no light. So most of the experiments and studies that I've read about is that they employ the 12-hour day, 12-hour light cycle. So it's just something to consider. So all these things you have to keep in mind and ask yourself, do you want to change to have the pH variability in your tank for this experiment or not? You just want it constant because it's not a key driver for you. So yeah, just keep that in mind, and it's your decision as well based on your research question. So in terms of treatment levels, so once we identify our driver, and then we say, all right, we want to test the response of this organism to ocean acidification. So our driver will be CO2. So, okay, how many treatment? So that's our treatment. So how many levels of CO2 do we want? Do we want just the current conditions, and then the very far future, which is like the end of the century scenario under IPCC? Do you want just those two, or do you want two more in between the now time and the future time? Or do you want another treatment where you also look at the pH from pre-industrial areas. So all of these things you have to create your treatment levels. How many levels of your driver do you want, or your treatment? For example, we can have pH now 8.03 and versus future. So we had two pH treatments. For my experiments, there were four. So there were four pH treatments. So it was ambient, a future, and then two in between. In terms of replication, it's also really, really important. I'm sure all of you are aware by now. When you're running experiments, you must have enough numbers of, samples, right, of organisms. And these treatments, replicates and samples must be independent from each other. So that's all part of the design and the setup as well. Again, you check your logistics, because if you want to have, 12 replicates, for example, for each of the species that you're working with, the question is, you go back to your logistics and ask yourself, do I have enough tanks to hold 12 replicates per species of the 7 species that I have? Do I have enough room in the lab to do that? Or should I just reduce the number of replicates? Again, you go back to your experimental design. So this is an example of why proper experimental design and setup and replication is important. So this is from a review paper by Boyd et al. 2018. And so it's just an example. So figure A, it's showing the response curve for 3 species that were under 3 different treatment levels. So 3 levels of the treatment, right? So it was 280 ppm in the CO2. So this is like pre-industrial pH, and then 400 ppm, that's like current conditions, and then 750, that's like future. So if you look at the raw data, like these points here, the data points, they seem to be doing the same thing. They seem to be showing the same response, right? If you just take the points. But if you look at the lines, the response norm, you notice that they are actually kind of different response, right? If you join the dots, for example. So, and that's 3 treatment levels. So for figure B, it's also a similar example. But if you notice here, there's like some of the experiments representing by different colors. They have 5 points, and some of them have 3 points, some of them have 4 points. So this is indicating the number of treatment levels. Now, one of the things is have more treatment levels or less treatment levels, but the other thing that's important to keep in mind is is the difference between your treatments. So, for example, if you look at the orange yellowish line here in figure B, it's showing 3 points, so 3 treatment levels. But if you notice, treatment 1 and treatment 2 are quite close to each other, and then treatment 2 and treatment 3 are quite far from each other. So the response norm is responding as such. If you notice the green line, the curve, the plot in green, you see there's 5 different time points, and the response is a bit different, right? Because they have more treatment levels, and I think they're a bit more equally different in terms of the difference in the treatment level. So all these things are important to consider when you want to run a proper lab experiment. So here's an example from my experiments. So this is an overview of the setup of our ocean acidification experiment. So this is what we wanted the physical setup to look like. Yeah. Oh yeah, sorry, I'm just going back to this slide here where it's showing, it says here that when we manipulate the experiment, we also have to have different tanks, okay? So they're called different things. So for example, these blocks here in between, they represent header tank. So header tank is like a mixing tank. That's where the water from your source, from out in the field, goes into the header tank, and then you bubble in CO2 or something, and then the mixing is done there. So that's your treatment. And then the water from the header tanks go into the experimental tanks. So this is for our ocean acidification experience, how we wanted it to look like. This is the actual physical setup in our lab. So if you see these pictures, we have the CO2 gas bottle here. We have these solenoids, and that's distributing the CO2 from the tank into the header tanks. So these are white tanks here, we call them header tanks. And then from the header tanks, the water flows into our experimental tanks. You also notice a lot of cables and electronics and everything here. So it can get a little bit more complicated given the number of species that you have, the number of drivers, the number of replicates, all these things will complicate your system. Or you can also have a simple system and have a simpler setup where you don't have a lot of replicates, for example, or treatment levels. So for us, for our ocean acidification experiment, for example, this is our treatments and our replication logistics. So we had 4 pH treatment levels: 8.03, that's current conditions here in Wellington; 7.93, 7.83, that's in between; and then 7.63 is what is predicted into the by 2100. So there were 12 replicates tanks per 4 treatment combinations, and there were 12 replicates per treatment combination per species. These replications are also important when it comes to the statistics when we analyze them, and so we have to consider that. Thanks, Kaylin. That's the end of that first part.
Thank you so much. That was fantastic. I feel like, I've run some OA experiments in my day, but if I'm ever going to do that again, I'm going to come back to this talk and make sure that I've considered everything that you've laid out in front of us. It's incredibly helpful. So thank you. Just a reminder, if you have any questions, feel free to pop them in the chat. We can go ahead and address those. But I'll share a little bit about another way to study biological responses to oceanification by basically looking at all of these studies taken together. So we had mentioned just how much time can go into running an OA experiment, and I would definitely not underestimate that, right? There's the organism to take care of. There's always something going wrong in the experimental setup too that requires daily care. So if you don't have a wet lab, or you may not have that sort of capability, a meta-analysis is sort of a neat way to be able to contribute to the OA field and learn about biological responses. So when I first started as a graduate student, I really didn't know what this was. I got it confused with a review a lot. But basically, a meta-analysis is taking all the research studies that have been done on a topic, so defining the parameters or the zones that you're interested in, and then using statistical analyses to sort of look at an overarching effect of those sorts of studies. So it requires just a computer, internet access. Subscription to journals is really helpful because you're pulling all these papers, although you can always write to authors and ask them to see the papers, and then some statistics knowledge too. But there's a lot of helpful resources out there for learning how to do meta-analyses. And so I'll just walk through the process really quickly. The first step is sort of defining what you're interested in. So if we're thinking about oceanification and effects on different species, we might start searching literature databases to find all of the potential papers that are dealing with that. And so that involves creating something called a Boolean string to search through these sort of web bases. So we might search for oceanification research, and then sorts of behavioral responses or chemoreception. All these little asterisks are showing that that word could end in a different way. So you could get chemoreception or chemoreceptor, for example. And so you basically plug this into a database like Web of Science and get back a number of different papers that are relevant. And then from there, it's really just a process of paring down to find exactly what you're interested in. So there's sorts of tools to do that, to look through different papers, including this one called Abstractor. So if you're interested in learning more about meta-analyses, we can touch base later, and I can show you all these different tools that are used. But once you found the subset of all the research papers that cover the topic that you're interested in. It's just a process of gathering the data back out of them. So there are different ways to look at graphs such as this bar graph here on the screen and actually pull out the exact value. So, our control may be 17.6 or, the pH treatment here may be responsive 16.1 or something like that. There's ways to precisely get that data back out. So then you as a researcher who's doing this meta-analysis has a whole bucket full of data from all of these different OA studies that then you can pool together and do analyses on and basically calculate the overall effect of different conditions. So you could say that I want to learn about ossification effects. I'm just going to say I want to look at studies that studied conditions at the end of 2100 and sort of bucket them that way. Or you can look at pH 7.9 versus 7.7 versus 7.5 and study responses in that sort of manner. So folks have been doing meta-analyses and for many different years. It's really popular in the medical field to understand, what the effect of a certain treatment is or a certain medicine. But there's been some work in the oceanification field as well. So I just want to share a little bit of that. Probably the classic paper is this Croker et al. 2013 paper called Impacts of Oceanification on Marine Organisms, Quantifying Sensitivities and Interaction with Warming. And this is a classic. I'd say that's pretty well cited. It is not in the reading material for this course, which I just realized. So we should probably get it up there because it's a really good resource. And so what I'm showing on the screen here is just a couple of figures from this paper that is a meta-analysis synthesizing all the work done so far on many different marine organisms. So along the bottom here and along the side here, you can see these different groups from calcifying algae, echinoderms like sea urchins, all the way to non-calcifiers like fleshy algae or fish. And then here on the y-axis is the mean effect size. So the effect size of future oceanification conditions on these organisms. And so an effect of 0 is no effect. So animals didn't do anything different in those oceanification treatments than they did in the ambient treatments in these experiments. A positive effect size means they responded positively. So for example, fleshy algae were shown to grow a little bit more under oceanification conditions. Whereas negative effect sizes indicate a negative response. So for example, coral calcification is decreased under these oceanification conditions. So this is just one example of a paper. A couple of years ago, I was working in Hawaii where we were developing parameters for an Atlantis ecosystem model, which basically incorporates a lot of different environmental information and sort of outputs it in a way that's useful for fishery management or marine spatial planning. But we wanted to add in the effects of oceanification and ocean warming. And so to do that, we got lots of data from lots of different papers around the Pacific. And so I won't actually go into like the results of what it showed, but I just want to point out a couple of interesting points. So this is a lot on this figure, but these are all different functional groups of organisms around the tropical Pacific. So sharks or coastal pelagics, filter feeders like sponges or bivalves, different types of fish groups, micronectin like zooplankton or jellyfish, benthic inverts like sea urchins, nutrient cyclers, this is a little bacterium right there, primary producers, lots of different functional groups of reef fish, and structural benthic species, which include groups of coral, like Poeciloporas right there, Porites, there's a couple different groups of those. And I studied both pH and temperature in this meta-analysis. You'll see two different colors. And this figure is basically counting up how many different studies there were for these different functional groups that I found through going through that process I just described of paring down the literature to those most relevant studies and then pulling the data out. So there's a relatively higher number than some other functional groups for primary producers, especially with oceanification. So studying macroalgae and macrofleshy algae right there. But there's some functional groups that don't have a lot of data at all, such as these sorts of fish. So that means that researchers haven't taken them back into the lab and studied them in a meaningful way to understand how they're going to respond to our future ocean conditions. That might be because they're maybe hard to collect. maybe they're too big to keep in the lab. That's a experimental consideration is as Louis was talking about, is how many tanks can you fit to increase your sample size. If you have a really big fish, you need a lot of space in order to run those sorts of experiments. And reef fish are also relatively understudied as well. So there's quite a bit of work on these planktivores, and these tend to be clownfish. So there's been a lot of groups around Australia that have focused on clownfish in the lab, and that makes up a bulk of the studies. So there's definitely still some key gaps in our knowledge and understanding of how organisms in the Pacific are going to be responding to these future ocean conditions because we haven't carried out these sorts of experimental studies. And I mapped where the organisms were collected to in the Pacific. So again, the blue colors pH studies and the orange colors temperature studies. You can see there's a really high concentration around Australia where most of the fish are studied. French Marais has research station there. But there's still a lot of areas too where we could benefit from understanding more about the biological responses of species. So part of the hope of this course, is that in a couple of years, we'll have much more dots to place, around this area in the Pacific and increase our understanding of how we expect organisms to respond to oceanification. And so now we're going to talk a little bit about some of these results from biological experiments, so you can hear a little bit more about what we found. Louia, do you want to take it over from here, or do you want me to start?
Kaylin, yeah, you can start.
Okay, so we know that based on these sorts of meta-analyses and just looking at the trends and coming out of experiments that not all species will be impacted equally. We looked at that Croker meta-analysis where it showed some of the algae might respond a little bit better, but we see a lot of negative impacts in some other groups of species like calcifiers. So it's important to consider not only the responses on growth and survival, but some of the functional effects as well. Perhaps an organism does really well, it can grow and survive in these future conditions, but its predator does really well too. And so that can be a challenge to sort of parse out. And then we're also thinking about physiological impacts and what those mean for species and ecosystems. I'll turn it over to you.
All right, thanks, Caitlin.
Would you like me to continue sharing?
Okay, I'll just. Okay, yeah, we can continue like that.
Okay.
Yeah. So here's an example of an experiment that we did and the responses, that we measured, for example. So as Caitlin was mentioning, and I saw the figures from her meta-analysis, that I saw that primary producers, research is going up, right? But it's still not a lot as compared to other organisms as well. So the thing with the seaweeds is that there has been good amounts of evidence to suggest that ocean acidification, for example, is detrimental for calcifying organisms. So that's something that has been, researched and has been supported quite well. There's also, like Caitlin said, that some species of some organisms might not be affected by ocean acidification, for example. So in the case of fleshy seaweeds, there's a bit of a mix in their responses. And one of the reasons is because of the variability in the responses of these organisms from different experiments by different groups. They usually provide, they usually show that variability. And so bringing that attention back to some of our experiments here with the Calerba, it's the green seaweed. One of the reasons that could attribute to the variability in their responses from other studies is because of their different inorganic carbon physiology. So one of the experiments, so my study and my interest is in the responses of caulerpa with different dissolved inorganic carbon uptake mechanisms to, warming, ocean acidification, and light. So what we did was we ran an experiment, a single factor type of experiment, where we tested the single drivers of ocean acidification and warming, and then we combined all these things together into a multi-stress experiment. So I'll just share some results from those experiments. So next slide, please. So here, what's interesting about these seaweeds is that, so all seaweeds in general, they need and they use CO2 for photosynthesis, right? But some species of seaweed, they can uptake CO2 from the water. So CO2 from the water, and that doesn't take any energy because it's passive diffusion. And then other species, they have what is called a CO2 concentrating mechanism, or CCM. And that mechanism allows them to actively uptake bicarbonate for, as a source of dissolved inorganic carbon. So that will cost them some energy, right? So we have two groups, CCM seaweeds, and we have non-CCM seaweeds. Next slide, please. So here, I'll just try to briefly explain this slide. It can be complicated, but just by looking at it. But this is a measure, one of the ways that we can measure the CCM. If a species has a CCM or does not have a CCM, is by measuring their carbon stable isotopes or their delta 13 carbon values. So you see that line in the middle, around -30. So all the organisms that are underneath that line, they are what is called non-CCM species. And those species rely exclusively on CO2. So that's their dissolved inorganic source, is CO2. And that's through passive diffusion. So the organisms that have delta 13 values above the green line that you see up here, they have a CO2 concentrating mechanism. That means they take up bicarbonate, right? So that's a measure. So we can run an experiment and then collect our samples at the end of the experiments and then prepare them for, some isotope analysis to give us something like this, like data like this. So what's important to note here is that for those seaweeds that do not have a CCM, that are called non-CCM, currently in the current CO2 conditions in the water, they are currently limited by CO2, right? Because there's not a lot of CO2 in the water. Now, projecting into the future, we are projecting under IPCC that CO2 is going to increase into the future, so pH is going to go down. So as CO2 increases, we are expecting that the non-CCM species will benefit from ocean acidification compared to the CCM species. CCM species take up bicarbonate and requires them to use some energy, right? So just by understanding that, we also know that from literature, the non-CCM species currently are rare in some of the ecosystems. They are kind of rare, so they don't usually report a lot of non-CCM species. There's quite a few studies on CCM species, testing them under ocean acidification and warming, for example, but not a lot of studies on non-CCM species. So it's interesting because if we are predicting that CO2 is increasing into the future, the non-CCM type species will maybe become more abundant, right, and more diverse. So next slide, please, Caitlin. So here in New Zealand, for example, there's two common species of caulerpa here in Wellington. So we have the sea grape-like looking like one that's on the top. So that's called Caulerpa geminata. And that seaweed, caulerpa seaweed, has a CCM. So the other common seaweed, caulerpa seaweed here in Wellington, does not have a CCM. It's called Caulerpa brownii. And so it's really interesting. And I think I am fortunate to have these two species commonly found here. And also they have different dissolved inorganic carbon physiology. So I wanted to use this as my model species to test the effect of ocean acidification on the green seaweeds. More on the seaweeds here in New Zealand, one of the species has a more northern distribution, so it has a more warmer edge into its distribution, and the other species, which is the non-CCM species, it is distributed more to the south of New Zealand, so it has a cold, range in its distribution. But these two species, they overlap here in Wellington. Thanks. Next slide, please. So here's a result, for example, from the experiments that we ran. Here's an example of a result for the growth responses for the OA experiment. So this was a single factor type experiment where we controlled everything else and just manipulated just the pH. And so we found that the non-CCM species, as you can see here, they kind of increase in their mean growth rates and also in their variability in their responses. You can see here under ocean acidification, right? So the colors mean from blue to red, it's current conditions. Red means really, really acid, like bad conditions or worst case scenario. So that's the non-CCM species. The CCM species looks like it's not doing so well, right? And it's a lot of variability in its response under the ocean acidification, which is the red bar. And remember the CCM species uptakes bicarbonate, right? So with increasing CO2, we see that it's kind of playing differently on these two caliber species. Next slide, please. So here's the growth responses from our multi-stress experiment. So this is a combination of all the factors that we looked at before in other experiments. So this is a combination of pH, or ocean acidification, warming or temperature, and light. And this is what we found for the non-CCM species on the left. We see that, because this is a multi-stressor, so if you see here in this second bar, it's looking like it's increasing in its growth rates under future ocean acidification. However, under the future conditions where light was low, warming was high, and pH was also high, it looks like this non-CCM species doesn't really like the warming so much. Although there's that increased CO2 under ocean acidification, it still doesn't like because of warming, right? And also this species is more distributed to the south, which is more cold range in its distribution. So it makes sense that it responds like this. The CCM species growth rates here. So it looks like it's increasing its growth rates under future conditions, so in the red bar here. So what's interesting to find out here is that despite warming and low light, it seems to be saved by ocean acidification. It's alleviated. The negative impacts of warming and light and low light is alleviated by ocean acidification for the CCM species. Next slide, please. So here's an example of the results from our carbon stable isotopes for the OA single factor type experiment. So for the non-CCM species, you can see here. So I tried to explain that before. So we have this reference line about minus 30. So any value below minus 30, it indicates that an organism does not have a CCM. So it makes sense that the Columbia brownia is not really, changing, right? Because it's already relying on CO2, and then CO2 is increasing in the experiment. So it doesn't really, it's just doing so well. It doesn't really change. However, for the CCM species, you see that under future ocean acidification, the delta 13 value or isotope value is decreasing under ocean acidification. So it's an indication that the CCM species is down-regulating its CO2 concentrating mechanism. And so it's starting to save some energy and now using CO2 that is now more available, right? So it's showing because we can identify that because it's showing that its value is going toward the minus 30 line. So it's heading toward that reference line where we think it's relying on CO2. Next slide, please. So it's something similar as well for the carbon stable isotopes from the multi-stressor experiment. We see the non-CCM species is still, below the minus 30 line. So it's not CO2 limited anymore, right? So it's more CO2. So it's doing all right. The CCM species here, however, we can also see that it's an indication of down-regulating of their CO2 concentrating mechanism, going toward the future treatments. It's decreasing its value to go down, which means that it's saving some energy, not uptaking bicarbonate, but just using the more available CO2 or something like this. And then the next slide, please. So what does all of that mean, for example, right? So after we do that, try to analyze, and then we say, okay, what does that mean at the end? So for the growth responses from these two experiments, it shows that the non-CCM species is indicating high reliance on the CO2 and the future ocean acidification. For the CCM species, it looks like ocean acidification, alleviated negative effects of future warming and low lights. In terms of their dissolved inorganic carbon physiology, what these experiments told us or is indicating to us or suggesting is that the CCM species shifted their DIC source. So not using bicarbonate, but now maybe going toward CO2 use. CCM species decreased their delta 13 values under future treatment, so that also makes sense. So overall, what this means in terms of the ecosystem. So if non-CCM species that we found respond positively to OA, then the effects of subtidal ecosystems could be larger than previously thought. Because now there's not a lot of non-CCM are not common, at least from literature. They say that's not so common because they're usually in the deeper water, more cold, low light conditions. But if CO2 is increasing into the future under OA, these non-CCM species will start to expand their distribution range, and there we might see a lot of them in the shallow regions, highlights, and yeah. So it's possible given the trend that we saw from the experiment. Thank you, guys.
Thank you so much. I think that's just so interesting to think about the two species of seaweed, right? In the meta-analysis that I showed, we generalized it all the way up to a functional group and then studied responses that way. But your work is showing, like, even within the same genus, we can have quite different responses to these sorts of future conditions. We have a couple minutes left. We do want to share just a couple of resources that we think are useful. Again, if there's any questions, feel free to put those in the chat, or you can raise your hand and ask them directly. We can also follow up as well after the talk today. So just a few things to point out in terms of analyzing experiments, getting to those results is really exciting after you finally finish up all the time in the lab. A couple different ways to learn more about doing that is this new course on data management and visualization that came out. It's open access, and I will share a link as a follow-up to this, but it's with PICRIC as one of the partners on this. So it uses data from the Pacific and goes through how to basically visualize it and work with it, which I think is really interesting. And then something that I think Sam has talked about in the lectures, but is a really good resource, is METAL, which walks you through doing research experiments and thinking about all the different aspects to consider. So thank you again for joining us all today. Again, if you have any more questions, we can answer those. If not, I'll give it a few minutes and we can wrap up. Oh, we have a question from Katie in the chat. Is this paper available yet? You're on mute.
Yeah. Oh yeah. Hi, Katie. It's on the way, so hopefully it can be out there soon. It's so hard taking my time on the revising the manuscript.
I think we can all sympathize with that. I'm still getting out my PhD research from years ago. Great. Well, thank you all again. It was really wonderful to have you here, and special thanks to our guest today, sharing she does have a lot of expertise, obviously, on this work. So many different great considerations to take when designing these sorts of experiments. So just as a reminder, we'll make sure to put this recording on our YouTube channel, and I'll make an announcement that when it's available sometime tomorrow. This week, we're also hearing from Dr. Sam DuPont on Friday at noon Fiji time to go over some questions that are in the discussion forum. So if you have been going through the lectures, anything pops to your mind, or you just want to pick his brain more about running experiments too, there's an option to join that and then get them answered directly. Next week, we're heading into module 6, which is really exciting. So chemical observations in the environment, and then from there, biological observations in the environment. So thank you again. Have a good rest of your day.